to say.
Yeah, don't do that.
You're positive for all of 1 minute.
Actually, you did
Okay, any questions, anyone? So tomorrow, lab number 3, you'll be doing street plates and pour plates, which are isolation techniques. You'll be getting your unknown. You'll be subculturing it into some broths. You can determine its temperature optimum. And we'll talk about the unknown report and the expectations and all that good stuff. Heck of it.
So can our lab notebook just be a regular notebook and we keep those in?
Yeah,
Okay.
absolutely. And like I say, I'm not collecting those. I'm not collecting for grading lab notebooks. So just however you want to organize your material, I think a separate notebook makes sense, particularly that way it gives you a place to put your unknown stuff.
Okay, thank you.
Okay. All right. So we're in the midst of talking about bacterial taxonomy, bacterial identification, bacterial classification. And unfortunately, in the prokaryotic world, the whole scheme of classification taxonomy is kind of a mess. And it's getting better. It's evolving. The ultimate tool that exists right now for both identification and classification is a publication called Bergey's Manual. And I want to give you a little bit of history about Bergey's Manual. We'll talk about how it's evolved and what various volumes are useful for and what they're not useful for. Mention a little bit about how you're going to be using it in identification of your unknown. And then talk about the most recent iteration of Bergey's Manual and how it's different from the very first ones that were published. So this is a good summary slide to start out with, giving you kind of a history of the evolution of Bergey's Manual. Bergey's Manual was first published, first edition, in 1923. The first edition of Bergey's Manual that was published was titled Bergey's Manual of Determinative Bacteriology. Since 1923, there have been 9 editions of Bergey's Determinative Bacteriology. The most recent one was done in 1994. All of those particular volumes are geared primarily toward identification of bacteria, particularly the earlier volumes, definitely up through the 7th edition. When you start getting into the 8th edition, it starts to get a little bit murkier because the authors of Bergey's Manual started to try to superimpose some taxonomy and some phylogeny onto the system, and it got really wieldy and pretty clunky and very confusing. And so I'll talk about how they address that particular problem a little bit later on. We only have the most recent edition of Bergey's Manual that we have is Bergey's 7th edition of Determinative Bacteriology. And that's this guy right here. And there are multiple copies of this. This is going to be the primary tool that you'll use for identification of your unknown. Okay? It basically consists of a whole bunch of keys. And the keys allow you, by choosing the correct choice based upon the results of tests on your bacteria, to ultimately the name of a genus and species of that particular organism. So very heavily kind of key driven. If I were me, I would make you use Bergey's Manual exclusively for identifying your unknown. That's problematic because to get it down particularly to genus, there are tests that you need to have information about to do the keying that we don't do. So what I've done is I have provided you with a shortcut to begin with, and that is in one of the appendices of your lab book, and we'll talk about that tomorrow. There is a key to genus for your unknown. And so I'm giving you an abbreviated key that will allow you to fairly easily get to genus for your unknown and not have to battle through Bergey's to get to that same point. Okay? Once you get to genus, then you can determine species using the keys in Berge's manual. Okay? So you'll have an abbreviated key to get to genus, and then once you've got your genera figured out, then you can do the keys in Berge's manual to figure out what species you have. Okay? The first 7 editions of Berge's manual, up to and including this one, rely primarily upon morphological characteristics of cells, morphological characteristics of colonies, biochemical tests like fermentation tests. So it's all of that information that really doesn't have anything to do with genes. So remember the kind of hierarchy: genes, proteins / enzymes, and then characteristics. Okay, so if you're looking at characteristics, you're not looking directly at the genes. You're looking at kind of the genes once removed. You're looking at the proteins that determine those characteristics and their reflection of the genes. So that dependence on those characteristics of morphology and biochemistry and stuff make it very difficult for this particular scheme to be very phylogenetic at all. It's primarily more of a practical identification scheme based upon those physical characteristics. So the first editions of Berge's manual tend to be primarily phonetic as opposed to phylogenetic. Okay, so the first editions of the determinative manual bacteriology are phonetic. Okay, now the 8th edition gets a little more interesting, and that's this guy here. The 8th edition, some of the genetics starts to creep into this, and this is where it starts getting a little confusing. So, for example, you will see no reference to GC content in the 7th edition or prior to that. They didn't really know about GC content at that time. In the 8th edition, now we start to see some reference to GC content. Another thing that you'll see reference to in the 8th edition is serotyping. And so, when you start getting to the Salmonella bacteria, there are going to be hundreds of serotypes listed in here, based upon antibody reactions to particular antigens that the different forms of Salmonella have. You don't see any reference to serotyping in the first 7 editions. Well, we don't do GC content and we don't do any serotyping in lab. And that's why the 7th edition is going to have the most useful information about your unknown. Now, does that mean this is useless, the 8th edition? No. You can use this as a verification, hopefully, of what you presume your unknown is, identifying it using the 7th. That's one of the mantras that I'm going to give you throughout the course of the semester regarding your unknown. The more different ways you can key it and identify it and come up with the same thing, the more confident you're going to be about the outcome. Now, all you're really required to do is to key it using the 7th edition. But if you're cautious, and I think smart, then you're going to try to verify that using the 8th edition, using the 9th edition, using whatever resources are available. And when I evaluate your unknown, if you miss it, if you've only done it one way and you miss it, that's probably going to mean more points missed than if you've done it several ways and come up with the same wrong answer. Okay? So that redundancy of trying to solve your problem in more than one way using more than one tool is not only a good strategy for getting the right answer, but if you don't get the right answer, you're going to get more points if you've done it that way. Okay, so we'll talk about that more a little bit later on. So, in this edition, the authors started to put some of that GC content, serotyping in there in an attempt to try to make this a little bit more phylogenetic than the previous editions had been. So it's kind of a baby step toward a little bit of phylogeny creeping into this. That's okay, people didn't mind that. But at some point in time they said, okay, this really isn't working for us as taxonomists, for example, or microbial ecologists. Our primary interest in these volumes is not just to identify things, we want to understand the ecology of an organism, for example. So, beginning in 1984, a new set of Berge's manuals were published. And notice that they've got a different title here. They're called Berge's Manuals of Systematic Bacteriology. And this was the first attempt to superimpose some phylogeny onto bacterial taxonomy to illustrate evolutionary relationships in the way these were organized. It took 5 years to get this first edition of Berge's Manual of Systematic Bacteriology published. There are 4 volumes now. Previously, there had only been a single volume. Why 4 volumes? Well, more organisms were known at that time, but more information was being included about these organisms. Okay, ecological information. Okay. We only have 2 of these volumes because the organisms you work with in lab only are found in 2 of the 4 volumes. The other 2 volumes contain some kind of obscure and organisms that we don't really refer to, so we don't really see the reason to buy those particular volumes. It's expensive. So these will be available, and we'll talk a little bit about the organization of Berge's Manual of Systematic Bacteriology. Well, okay. Then people who were trying to use Berge's Manual for an identification tool started to get kind of frustrated because they said, this is more recent, but this doesn't work nearly as well in identification as this does. That this just has too many purposes it's trying to fulfill. It's trying to be an identification tool, it's trying to be a phylogenetic aid, and it's not doing a good job really at either very well. Okay? They said, we want a good up-to-date identification tool. We don't care about all this phylogeny and ecological importance. We want to be able to identify bacteria in a very straightforward manner. So, in 1994, they produced the 9th edition of Berge's Determinative. So we're back to determinative again. Identification. Okay. Basically what they did was they took out all of the identification material from the 4 volumes of systematics and kind of abstracted it or condensed it into this 1 volume of the 9th edition of Determinative. So they basically pulled out all of the phylogeny and ecological stuff and condensed it into an up-to-date identification scheme. So can you use this for identification of your unknown? Yeah, you can. Now, the way that these 3 determinative volumes are organized is a little bit different. This one, the 7th, has primarily keys in it. And I think keys are very useful in terms of being able to identify organisms. It gives you a very systematic way to go through the identification process. The 8th edition still has keys, but then you'll see also in the 8th edition a lot of charts where they'll have various species, and they'll have all these tests, and they'll have all these results. I personally find charts harder to deal with in terms of identification than keys. But there's a greater emphasis on charts and putting information in tabular form than in the classic dichotomous key. And that really kind of continues in this 9th edition. This is more chart-driven as opposed to key-driven. Okay? So each of these tools have a little bit different philosophy and use. And I'd like you to look at all of them and consider all of them good resources for your unknown. But again, the primary one will be the Berge 7th edition. That's where you begin keying once you determine your genus from the genus keys I provide in your lab book. Berge 7th edition is available online. I think it's a little bit hard to navigate online, although you can search for particular species in that document. That's why we have hard copies, but if you're working on your unknown at home, if you just Google Berge 7th edition, it'll come up. And particularly if you're trying to compare various species within a genus, that's manageable on an electronic format. But in your first keying steps, I think it's very difficult to use the electronic version. I think that the hard copy of that is much more user-friendly. Well, then in 2001 and through 2012, the ecologists and taxonomists said it's time to update the systematic editions. Okay? Because it's been, since 1989, and there's been a lot of discovery since then. So in 2001 through 2012, the second edition of Berge's Manual in Systematic Bacteriology was published. And that is 5 volumes. The previous edition was 4 volumes. More information. One of the fundamental differences in first edition and second edition of systematics is that first edition was based upon 5 kingdoms of classification. The second edition was based upon the 3-domain system of classification. So what we're going to do is we're going to do a little survey of the microbial world and talk about some bacteria that you will either encounter in lab, might be your unknown, or that have some kind of medical or other really big importance. And we're going to see where that fits into the currently accepted scheme of classification. That scheme of classification being Berge's second edition of Systematic Bacteriology. So you totally confused now? Okay, so let's go through this kind of a step at a time here, starting with Berge's Manuals of Determinative Bacteriology, editions 1 through 8. Each of these are a single volume. And the organisms are organized into these volumes into 19 parts. Now, you might say to yourself, well, wait a minute. Here's our traditional Linnaean classification hierarchy. And then we've added domain, kingdom, phylum, class, order, family, genus, species. Where is a part in that particular hierarchy? There isn't such a thing as part, right? This is an artificial construct. And what happens is the authors of Berge's Manual, there's a Berge's Trust and a board and all kinds of people who are contributing to the publication of these particular volumes, got together and said, we're going to organize all of the bacteria that are known at this time into various convenient working groups. And we're going to get a set of experts who are in charge of each working group. And those experts are going to be responsible for organizing taxonomically those 19 parts, and how they're organized might differ from one working group to another. Seems like kind of a chaotic way to do it. And they tried to use some of the traditional Linnaean taxonomy to organize each part. But some of these parts, some of the working groups said, we're only going to have family, genus, and species in our working group, in our part. Others say, no, we're going to have classes, families, genus, and species. Others say, we're only going to have genus and species. Others say, we're going to have some groups that don't even have any taxonomic status, like other organisms. Okay? So as you start looking through Berge's Manual, you're going to see this difference from Part 1 to Part 2 to Part 19. That particular philosophy has kind of been retained in Berge's 9th edition. The 19 parts became 35 groups. Okay. In Berge's Manual of Systematics, both the first and second edition, there are 33 sections. But again, the idea is similar in that there are working groups of experts who get together and kind of organize the taxonomy. In the second edition of systematics, they're trying to get more consistent from group to group. So they're trying to move toward more of kind of a universally accepted means of organization. But this is kind of a carryover from the way the original editions of Berge's manual were originally organized. So these ideas of parts, groups, sections don't have any counterpart in the traditional taxonomic scheme. And that's where things get a little bit disjointed. Okay, so determinative bacteriology, 1923 through 1994, one volume each, organized into the 19 parts for editions 1 through 8. Edition 9, 35 groups. First 7 editions are primarily phonetic. A lot of morphological, biochemical information to organize these things. 8th edition, attempting to become more phylogenetic. That's where you start getting some of the clunkiness now and some of the displeasure with this, that it really serves neither purpose very well. So 1994, the 9th edition, goes back to more strictly phonetic identification. Berge's Manual of Systematic Bacteriology. Okay, the first attempt to do a phylogenetic classification. Published in 4 volumes, 1984 through 1989. Don't worry about the dates. These volumes are titled Gram-negative bacteria of general medical or industrial importance, Gram-positive bacteria other than the actinomycetes, Archaebacteria, cyanobacteria, and the remaining gram-negative bacteria. Remaining meaning they don't fall into this particular category. Medical, industrial importance. And then volume 4 are the actinomycetes. As I said, we only have volumes 1 and 2 because all of our organisms we work with in lab will either be gram-negative bacteria or gram-positive bacteria with some kind of importance. Okay, this one's a little bit hard to see. So let's dim the light for a second and maybe try to zoom a little bit. Okay, here are our volume names, volume 1 through 4. Again, I just went through those. These numbers in parentheses indicate which of the sections are found in those particular volumes. So volume 1 has section 1 through 11, volume 2 section 12 through 17, etc. Don't worry about memorizing those numbers. Okay. They're just there for illustrative purposes. So just in this example, so you can see kind of the inconsistency that there is sometimes from section to section and how they're organized, take a look at volume 1. So these are gram-negative bacteria of medical or commercial importance. And the first section in volume number 1 are the spirochetes. Look how they're organized. There is an order with 2 families. This family has several genera. This only has 1. And then there is this catch-all category of other organisms. Okay, that's how the experts of section 1 chose to organize their particular organisms within Berge's manual. Okay, but look at section 2. These are the aerobic / microaerophilic, helical, vibrioid, gram-negative bacteria. Do you see any orders in there? No. See any family in there? No. They are just a group of several genera. Okay, so they have taken a different taxonomic strategy, if you like, to organize theirs. Okay, section number 3. These are the non-motile or rarely motile gram-negative bacteria. A single family with 3 genera, but then they've got this other catch-all category, other genera. Okay? So really, what I want you to really see here is just this illustration of how there is a difference in how these working groups chose to put things together and try to superimpose as best they could traditional Linnaean categories onto this identification system. Is that?
On the section 2 where it says gram-negative, what is that mistype or is it correct?
Oh, it's a mistype.
That is.
Yeah. Should be gram-negative. No one's ever seen that before. What they tried to do when they put together the first edition of Bergey's Systematics was to superimpose as best they could some traditional Linnaean taxonomy onto this system. So they decided that there would be division names assigned to various types of bacteria. So for example, the gram-negative bacteria were called division Gracilliichides. Division number 2, the gram-positive bacteria were called Firmicutes. Division 3, Tenerichides. Division 4, Mendocichides. And then under those divisions were identified various classes. If you want to see where this kind of fits in with traditional taxonomy is, sections kind of fall between class and order. So if you're doing identification of this particular bacterium, Borrelia burgdorferi, it was assigned to the kingdom Prokaryote, the division Gracilliichides, the class Scotobacteria, and then they were put into section 1 of Bergey's manual, and then section 1 is divided into orders, families, genus, and species. Okay? So even though these particular sections don't really have official taxonomic status, they kind of fall in here operationally between class and order. What is important to know, and what makes this kind of confusing, is you might think that the 4 divisions of bacteria would correspond to the 4 divisions of Bergey's manual. They don't. Okay, so that's what these Roman numerals over here indicate. Okay, so division 1, the Gracilliichides, gram-negatives, you can find those in both volume 1 and volume 3. The Firmicutes, yes, are only found in volume 2. The Tenerichides are found in volume 1. The Mendocichides are found in volume 3. So there's an overlap between these division names and which volumes they fall into. So just understand that there is no correspondence between these division names and volumes of Bergey's manual. So that's just kind of background, okay? Really, this is where the rubber's going to kind of hit the road in terms of you being able to place organisms we're going to talk about and encounter in the class into a system of classification. And that will be Bergey's Manual of Systematic Bacteriology. And we do not have these hard copies, okay? They're incredibly expensive, and they're really not of any use for us identification-wise. If we taught microbial ecology courses and some more advanced taxonomy courses, we would have these. But they really don't have any purpose for this particular lab because they're not intended to be identification tools. Okay? So here are the 5 volumes of Bergey's Manual of Systematic Bacteriology. And this is kind of the landscape in which we're going to insert and superimpose the organisms that we talk about. And at the end of these PowerPoints, I'm going to have a little summary slide for you that basically says, what do I need to know about taxonomy? Okay? And you'll see that it's not as intimidating as it looks right now. So, volume 1 are the archaea and the deeply branching phototropic bacteria. These are some photosynthetic bacteria. Volume 2 are the proteobacteria. Volume 3 are the firmicutes. That name might sound familiar. That was one of the names of the divisions in the previous Systematics Edition 1. Volume 4 has a whole bunch of different kinds of bacteria in there, including Tenoricides. That used to be one of the division names in the previous edition. Okay? And then Volume 5 are the actinobacteria. So basically what we're going to do is we're going to kind of take a march through the 5 volumes and see where they fit and how some commonly and interestingly observed organisms fit into this particular classification system. Are they in Volume 1? If so, where are they in Volume 1? How is Volume 1 organized? So, this is a great slide because this kind of summarizes the 5 volumes. Okay? So here are the volume names right here. And then this shows you the domains that are found within that volume. It then shows how those domains are broken down into phyla and classes. Sometimes they're only broken down into phyla. They're not subdivided further into classes. But you see that the lowest level of traditional hierarchy found in these volumes that's identified on this slide is class. So it kind of breaks everything down from here to here. Couple of things to note. The first volume is the only one that contains 2 domains. This volume contains both the domain archaea and the domain bacteria. All the rest of these are found in the domain bacteria. So volumes 2 through 5 are the domain bacteria. Volume 1 is partly archaea and partly domain bacteria. Another thing to notice here is that there is a very high emphasis placed on GC content. So for example, here in Volume 3, we have the low GC gram-positive bacteria. In a different volume, there are the high GC content bacteria. Now, remember, this particular set of volumes, the second edition of Systematics, is really attempting to show phylogenetic relationships. So the fact that these are in different volumes, and therefore different phyla, indicates that there's a real fundamental evolutionary distance between low GC bacteria and high GC bacteria, even though they're both gram-positive. Okay? So just because you're gram-positive doesn't mean you're closely evolutionarily related. What's more important to show that evolutionary relationship is your GC content, which is really a reflection of your genetic composition. Does that necessarily reflect into recognizable morphological characteristics? Maybe, maybe not. There are 2 things that can look very similar morphologically, one of which has a low GC content, one of which has a high GC content. In past editions, they would have been put together because of their similar morphology. Now, in this edition, they're split out because they have very different GC contents, indicating different evolutionary ancestry. Does that make sense? Okay, so let's start with Volume 1 and domain bacteria and kind of look at a few of these things, and I'll point out a couple of them that might be of interest to you. Correct?
Right on the slide before, Volume 4 is listed as having bacterioputes, and then on the next slide, it's Volume 5.
Which one?
On this one, it says Volume 4. If you go to the next slide.
Which one were you saying was on Volume 4? Then?
Oh, all of these. Like, and you go over here, it's Volume 5 instead. Because Actinobacteria is volume 5.
Volume 5, yeah. Now this is from the textbook, and this is from Bergey's Trust. Well, we got to check that one out. Okay? There's an inconsistency there, you're right. For right now, we will use, since I'm going to have this slide plastered all over the PowerPoints over and over again, let's use this, and I'll go back and check the Bergey's Trust information and see if that is the one that's been misrepresented. Somebody's wrong. Good catch. Okay, so let's take a look at the domain RK. Sometimes the RKs are called the other prokaryotes, or more commonly they're referred to as the ancient prokaryotes. We think that these are some of the first organisms that evolved. Some, but not all of these, are characterized by living in pretty hostile environments. I mentioned the term thermophil. I mentioned the term halophil. Did I mention halophil? Those are organisms that live in very salty environments, like the Dead Sea. Kind of a general catchword for these organisms sometimes are the extremophils, the ones that live in very extreme, inhospitable environments. The RK are really interesting critters, and they really kind of upset that normal, clear dichotomy between prokaryote and eukaryote. They're kind of in between. They have some characteristics that are more prokaryotic-like. They have some characteristics that are more eukaryotic. Cell type, clearly prokaryotic. No membrane-bound organelles, 70S ribosomes. Okay. They have a single circular chromosome like bacteria do. While they do have a 70S ribosome, it's actually more similar in structure to an 80S than it is a 70S. And what's interesting is that antibiotics that will kill bacteria usually won't work against these RK bacteria, reflecting a little difference in their ribosomes. They share a number of RNA sequences with eukarya. Bacteria typically share one common sequence with eukarya of RNA. RK bacteria, 3. They actually have protein synthesis that is more similar to eukarya than to prokarya. They have a cell wall, but it's not made out of peptidoglycan. I think I mentioned before, sometimes they're found with protein cell walls, sometimes they're found with carbohydrate cell walls. Cell membranes, the lipids are a little bit different. Prokaryotic bacteria tend to have fatty acids with ester linkages. RK tend to have long-chain branched hydrocarbons with ether linkages. So there's this kind of weirdness where the RK are kind of eukaryotic-like in some respects and kind of prokaryotic-like in other respects. Here are a couple of examples of some interesting RK bacteria. These are organisms called extreme halophils. And here you can see these are retention ponds that have very high salinity. And you can see as the water evaporates, all the salt that's being left behind as the water evaporates. And these are highly, highly saline environments. Normally, most bacteria cannot survive in those particular environments because the environment is extremely hypertonic, and that sucks the water out of the cells and dehydrates them. But these RK have adaptations, including pumping solutes into their cytoplasm to kind of make themselves isotonic to the environment so that they don't lose all their water. Okay? Some of these particular bacteria produce various pigments. You can see the reddish pigment produced by this species. You can see a yellowish pigment produced by this species. You can see more purplish pigments produced by these species, but very unusual microorganisms. So in this domain, there are some bacteria that are found in this class, the halobacteria. That's the ones that we were just showing you right here. Those are halobacteria. Oh, are they in different order? One of the things I want to warn you to, and it's good we're coming up to time when we're about, the version of the lecture notes that have the corresponding slide numbers on them, that's both on the website, which is kind of working and kind of not, and on Dropbox, was an older version. So I'm going to go back after class. You're good through the first 20 slides. The correspondence is right. But after slide 19, I believe, the correspondence is off. And so I'm going to go back and update those. And so you may need to reprint those last 20 sections of lecture notes that refer to slides 20 on. Okay? Because otherwise the slide numbers will be off. And you all know you can print in here, right? Okay. So you don't have to pay if you print in here. The second type of archaebacteria are the extreme thermophils. And they are found in a genus right here, the thermococci and the thermoplasmata. Anybody know where this is found? This particular slide? Yeah, it's Yellowstone. This is Grand Prismatic Spring. And what you can see is that in the very center of the hot spring, the temperature is the highest. And then as you move out toward the edges, the temperature drops a little bit. And you can see different colors associated with the different species of bacteria that can inhabit those different temperatures. But even in these highest temperatures right here, which is boiling, there are species of archaebacteria that can tolerate those particular environments. Now, you have to kind of wonder a little bit about what adaptations do they have that allow their proteins to remain intact even under boiling conditions. Okay? One of the enzymes that's very important in biotechnology comes from these particular types of environment. There's a bacteria called Thermophilus aquaticus. And that produces an enzyme which is highly heat stable called TOC polymerase. TOC polymerase stands for Thermophilus aquaticus. And this is a DNA polymerase that is used to copy DNA. How do we use TOC polymerase in biotechnology? Anybody know? This is used in a really important technique called polymerase chain reaction, PCR. And PCR is a way you can rapidly duplicate or copy DNA. And the process involves heating DNA to separate it into two strands, and then copying the DNA, and then cooling it so it reanneals, and then heating it again so it separates, and you copy it again. You do this alternate heating and cooling process to copy DNA very rapidly and make millions of copies in a short period of time. Well, before they found this TOC polymerase, every time you did a heating and cooling cycle, you had to replace the polymerase because the heating process denatured it. Now, when you've got this heat-stable polymerase, you can use the same polymerase in cycles over and over and over again. You guys use the thermocycler, right, in biotech? Okay. So that TOC polymerase came from this archaebacteria, this extremophil bacteria that is adapted to live in these very, very hot type of environments. Also in volume 1 is the, a couple other things right here. Another group of these archaebacteria that are pretty important are a group of organisms called the methanogens. So you can see over here, there's a class called the methanobacteria. There's another class called the methanococci. We'll talk about this more when we talk about metabolism, but methanogens have the ability to combine carbon dioxide and hydrogen gas to make methane. These particular organisms tend to be anaerobic.
Are these organisms found in the rumen?
They are. They're found in the rumen of cows. And cows, as they digest grass and the bacteria that feed on grass, produce methane. And then they burp methane into the environment. Okay? Another place where you can find these methanogens is buried down at the bottom of layers of sediment and ponds. Have you ever seen any pictures, or maybe seen it yourself, of a pond where there are bubbles that arise? And actually, if you're floating around in a little boat there, if you have one of these, for example, when that swamp gas comes up to the surface, you can light it and burn it. It contains methane. Okay. Ecologically, what's the importance of methane? It's a greenhouse gas. Okay? And like carbon dioxide, it traps heat and contributes to increasing global temperatures. Rice paddies have anaerobic bacteria that produce methane. And so there are all these natural processes that produce methane in addition to landfills. Bacteria can produce methane in landfills. And there's a fairly significant amount of methane that accumulates in the atmosphere as a result of this particular bacterial metabolic process of methanogenesis. So we'll talk a little bit more about that in the context of bacterial metabolism later on. Okay, so where we'll pick up next time is we'll look at this domain bacteria a little bit. And particularly, we're going to concentrate on a couple of phyla within that domain in Volume 1. The phylum Cyanobacteria and the phylum Chloribia. And those are going to be photosynthetic bacteria. And there's two different strategies of photosynthesis that you find in the microbial world. There's oxygenic photosynthesis, like higher plants do, and there's the anoxygenic photosynthesis. I briefly mentioned it when I talked about sulfur granules and sulfur bacteria. So we'll dig into that a little bit more. So I will send out, I'll just do it by email, an updated version of these lecture notes with the proper sequence of numbers on them. If you've already printed them, you're, like I say, you're good through slide 19, and so you only have to print the rest of that from 19 on to be up to date. Okay? Make sure you read your lab for tomorrow.